Hey, welcome to Research Jam number 21. I was looking at the submission form yesterday just to get to know how many people submitted. So far we have over 100 people submitted to Research Jams from the start, which is not that big of a number considering we've been doing it for 3 years, but still pretty impressive. So thanks to everyone who have presented and the 3 speakers today who are going to present. We'll start with Francesco talking about something biology, mapping human transcriptomes to cellular proportions. That's your title.
That's the title. Yeah, I changed it a little bit, but yeah. So thank you for having me, first of all. And so I'm just gonna go ahead and share the screen so that we can be super efficient.
And Francesco, while you're sharing, I'll mention
There you go.
I'll keep track of time, and at maybe T-1 minute, I'll write something on the screen or just pop in to say we should wrap up.
Yeah, absolutely. I'm also timing myself just to be considerate. Okay, so thank you for having me. So just to give sort of like level the expectations, my background is in biology. I'm a computational biologist who's been dabbling in ML for different applications, and my goal in science and life is to actually do something really meaningful regarding the immune system. And the reason why I care about it is because the immune system is one of the most important and coolest components of the human body. It's involved in pretty much any biological process that is related to disease. And if you have one, you think, oh, there's nothing immune related to it, chances are that you're wrong. You probably just haven't found it yet. And the more people dig on this particular type of connections, the more they found. But one interesting aspect about the immune system is also that if you take this idea and you flip it on its tail, the immune system is actually a sensor. One of my advisors during my postdoc, Parvesh, used to say that the immune system is a distributed sensor with a signal amplifier built in on top. And it's actually, I would say, quite correct thing to say. Whenever there's something that happens in your body, chances are that your immune system is reacting to it, and so it's changing. So measuring the immune system, you can actually figure out what's going on inside your body. And so one of the things that has been done in immunology that is like sort of very, very, I would say foundational in the way, diagnostics work is actually measuring changes in the immune cells. And so you can take a blood sample and then simply count the different types of immune cells that you have. And oftentimes, changes in the counts that you observe that would be different compared to what you would expect to be a normal reference sort of like range often are associated to something like having, for example, infection. That's the case of HIV, having a bad flare of an autoimmune disorder, or, for example, the baseline level of immune cells can give you information whether you respond to immunotherapy or not if you have cancer. So all those things are really important, and this is basically sort of like how people think about immunology and the immune system. Obviously, to be able to do all of this, you need to be able to get samples and measure cells. And while this is usually the correct route, it has some limitations. Obviously, these are all these 3 things that you see over here. For those of you that don't know, are 3 machines that people use to take biological samples and basically count and measure aspects of the immune cells that you have there and count them. And obviously, this is the ideal way. You can quantify the cells directly. Sometimes you can even save them afterwards. You can do experiments on them. But the problem is that you're always limited by the number of samples that you put into this experiment. It's difficult to acquire them, so it's expensive. And oftentimes these experiments are not comparable to one another. You basically have, for example, different experimental types of experiments, different reagents that you use, and sometimes there's a subjectivity in the way these experiments are analyzed, meaning that comparison between different experiments is not always possible. And so this is basically how do you overcome the fact you don't have enough samples, how do you overcome the fact that you might not be able to really compare experiments between one another. So sort of like on a parallel track to all of this, we've been generating in biology a ton of molecular data. And oftentimes this data is released in the public domain through databases like GeoSRA, Links, Cell Atlas, etc. And this is a set of data that is growing at an exponential rate and is oftentimes underutilized. Most samples present there are samples where you profile whole tissue biopsies or blood samples as a whole. So you kind of like blended them together, and then you basically measure a lot of different properties of them, like all the proteins there, or all the genes there, or the RNA there. And the idea is that we reached a point where there's over $10 billion worth of samples that have been profiled in these databases, and oftentimes people don't use them in a very effective way. And so, the question is, can we use this growing body of data to actually learn something about the immune system by applying machine learning to it? So, the way I like to think of this is that bulk data is like a milkshake. So I love milkshakes. I did my PhD in the Midwest, and they love the milkshakes. Over there, they really love milkshakes. And if you want to do something like a good strawberry milkshake, you need to do strawberries, milk, sugar, and ice cream. And so I can give you a recipe. This is a little bit made up, so don't necessarily follow it, in a literal way if you want to make the best milkshake. But the point is that this is usually what people think about. You put the ingredients there, and then you blend it, and you make a milkshake. But what if we try the opposite problem, which is we get the milkshake, we taste sample it, and we say, okay, there's X amount of strawberries, X amount of milk, X amount of sugar, whatever, right? Can we guess the amount of each ingredient just looking at the blend? That's basically the question that we're trying to answer. And so we have a methodology in a computational immunology called cell-based deconvolution, which is basically biologists usually normally call in silico flow cytometry. The idea is that basically you have this sort of blend of genes and proteins that you're measuring. Think of it as a vector of different transcripts or different RNA, and you have a numerical vector where you say, okay, I have X amount of this gene, X amount of this gene, X amount of this gene, X amount of this gene. And what I'm trying to do is basically estimate the cells that were inside that sample that generated this pattern of gene expression you're actually looking at the end. And so this is basically, when you think about it, it's a linear regression problem where you have a single sample that you're looking at. You have a matrix of what sort of like gene expression or amount of RNAs that you have for each individual cell type. What you're trying to do is you're trying to find the linear combination, the optimal linear combination of each individual cell type that can generate the observed pattern of gene expression that you have in the sample that you care about. So if you think about it, this is basically, this makes it really interesting because then you can sort of like, in a very primitive way, map one type of data, which is basically RNA and proteins, directly into cell proportions. And so the beauty of it is that now you can tackle, if you get it to work, you can tackle anything that you have in the public domain. And again, just to give you an idea, it's basically, you can think of it as a linear regression problem where basically it can be a linear regression. There's many others that people have tried. But the idea is that you start off with vector gene expression, you want to get a vector of cell proportions in output. And so the question is, given that there's so many samples out there, is this sort of like this approach generally robust across samples that have a lot of heterogeneity in terms of both biology as well as sort of like technical differences? Because every sample oftentimes different samples oftentimes are measured with different protocols and whatnot for experimental protocols, experimental technologies. And so we decided to tackle this question by basically doing an analysis where we look at over 1,000 human samples across 17 studies and 8 microarray platforms. For those of you that don't know anything in terms of biology, microarrays are just the technology they use to measure basically levels of gene expression. And so we measure, because it's a linear regression, we measure goodness of fit of the regression, basically just the correlation between y and y-axis in the regression. And we use it as a proxy of the convolution accuracy. And because there were two matrices, going back to this, because we have a linear model that basically uses a matrix of like what is the expected patterns of gene expression per cell type. We call that basis matrix, and we use a regression-based algorithm. Those are basically the two variables. And so there were two matrices of cell, sort of like gene expression signatures between different cell types. one is called Iris, one was called LM22, and 5 different algorithms that basically can be used. There are different regression algorithms that can be used for this type of approaches. And what you can see is that in this particular plot, we have different levels of accuracy. Most of it is pretty low. Everything looks pretty much red if you look at it. But there's also a lot of differences between different columns, which represent different sort of experimental platforms that have been used to measure the levels of RNA molecules for each of these samples. And so what you would expect to see, yeah?
Can you remind us what are the elements on the x-axis here? Each bar.
Yeah, absolutely. So the x-axis is basically each group represents a different algorithm. Each sort of like a plot. Each plot on the rows is different basis of parameters. Each column represents a set of samples that have been profiled with a particular platform, a microarray platform. So there's 8 of them, and sort of like the bar plot that you see there. Each sort of like the fraction represents the fraction of samples that have been sort of like you've done this deconvolution method, and sort of that represents the accuracy of it. That's the distribution of accuracy scores for each sort of deconvolution experiment.
Okay, that makes sense. And then for each bar, do you actually have ground truth, which is how you're computing this goodness of fit?
We'll get to that, but for all these thousands, but we have a set of 400 samples for which we do, and I'm going to show you this in a few slides. But that's a very, very good question. So ideally, what you expect to see, everything would be basically blue across every method, every matrix. That would be the ideal scenario. As you can see, most of it is red, but there's also heterogeneity between the different platforms. So the question is, to see this, so we started to do a little bit more digging, and we decided, okay, let's compare accuracy by comparing sort of what would happen if we use a different method or what would happen if we use a different matrix. And we simply did analysis. What we ended up finding was that the regression method that you used actually didn't matter as much. Really, what mattered was the matrix of parameters that connected the gene expression data to the cell type. And that, we figure, was the major source of confounding. So we decided to design a new one that would basically be potentially more robust to confounders. And just to give you the idea of what we mean in terms of robustness is that we build these things, we actually try to integrate as much data as possible, specifically multiple independent studies. And for example, if we want to find a gene that is really relevant for a particular cell type, we want to find a gene that is high in that cell type and low everywhere else across multiple studies. And so hopefully, that's basically the situation of the gene X, whereas gene Y is not consistent across all these studies. So it doesn't look like it could be a very good biomarker. And so we basically integrated 6,000 samples from the public domain, from the set of public data that I was telling you about, normalized it by scaling and quantum normalization, and we calculated basically fold changes through effect sizes. And the idea is that we used all this data to basically do a feature selection where we identify roughly 15, 20 genes per cell type. And basically that result is generating a matrix of gene, sort of like gene markers for 20 different immune cell types that we call immunostates. And so just to give you a quick sort of overview, we found that immunostates actually worked a lot better, except for the PERT algorithm, which really sucked no matter what you use. It actually really, it's not perfect, but it's a lot less confounded, significantly less confounded. I'm not going to go over details of other tests that we've done, but this is what Jason was suggesting, the ground truth experiment, where basically we had a set of 402 samples for which we have both the gene expression data as well as the ground truth measure by counting the immune cells present on the samples using a technique like flow cytometry, for example, or mass cytometry. And what you see is that using this approach, irrespective of regression algorithm that you use, you actually see that there's a higher correlation compared to the ground truth compared to the other methods. And that's likely because you're being more robust, less biased, less confounded, and therefore more likely to get to being accurate. And so
If
that's basically what we found.
we
Yeah.
should wrap up in about 1 minute if you can.
Yes, we absolutely. I'm basically towards the end of the talk. Excellent. Thank you for the reminder. Okay, so I just want to give you a quick application, which is basically we apply this to different problems. One of them was TB, tuberculosis. Tuberculosis affects one third of the world population. We're trying to figure out what would change in the immune system throughout different phases of the disease. And I'm not going to go into details, but basically this is a published work, but we apply this approach to over 1,000 human subjects from public data. By taking cell proportions and integrating it, we actually found two cell types that change in tuberculosis from the alpha state to the latent phase of the disease, and from latent to active disease. There was an immune cell called NK cells. And lo and behold, we actually validated two independent cohorts, one from South Africa, one from Taiwan. We published this paper in Nature in 2018. If you're curious, these are the references. You can take a look at them. We apply this to other problems as well, like flu, pulmonary fibrosis, etc. And this type of approaches actually works really, really well. And with that, this is all I have to say. I just wanted to give you a quick overview. These are the people that contributed to all the work. And I'm mentoring currently a student in one university in Italy, and she provided this beautiful illustration of the immune system on the right. And at this point, this is it. That's all I have to say. So thank you for your attention.
Thanks for presenting. That's really interesting.
I hope it wasn't too biological or too domain specific for the audience. But if it was, you can ask me any questions right now.
Yeah, no, it's definitely outside the domain of many people here, but it's obviously interesting to see machine learning being applied to these other areas. We should probably hold questions until after, just for the sake of time. Maybe we should get to the next person. And Francesco, are you on Discord? There's a channel on Discord for the jam. Maybe we can do questions in there.
I think, yeah, that would be great. At 8:45, I have to take off. So yeah, if I'm not here by that time, I'm happy to take the questions afterwards on Discord. Thank you.
We'll move on to our next speaker. Thanks, Francesco. Suman.
Do I start?
Yes.
Okay. First, I would like to thank MLC for giving me this opportunity to present, and also everyone who supported during my research. Okay, so it is long ongoing research, I guess, for me, because I was trying to find locally activating neuron and what that means. I'll get into it shortly. Okay, so first, when we think about neural networks, basically an MLP, like something like this, come into our mind, right? Okay, so something like this, and there might be confusion to beginners because it doesn't give every information about what is going inside here. Like there are bunches of numbers there. It is optimized with gradient descent, but what is exactly going on? What do each of these neurons represent? These are not easily understandable. So I was trying to understand how to create interpretable neural networks or understandable neural network in terms of neurons. So how can we understand individual neurons and what they are doing? And to get started, I'll start with a simple one-dimensional regression problem. So this is a non-linear regression problem. And if we are using one-layer ReLU MLP with ReLU functions, ReLU activation functions, then multiple ReLU neurons are represented by this blue line. So this is one ReLU, this is another ReLU, and yeah, so multiple ReLUs join together to form a highly nonlinear function. And the problem is that these two neurons, they superimpose with each other, like they share a region where they activate, and some neuron activate everywhere, mostly everywhere, and it is difficult to uniquely identify a reason for a neuron, because these neurons are global. And if we see this two-dimensional regression, here also multiple pieces of ReLU add together to form highly nonlinear function. And we can see simple visualization that if in the same direction of ReLU, like similar to here, if it is farther away from decision boundary in the positive side, then it activates highest. And multiple neurons activate for the same input. And it is difficult to find out what causes it, like what is the main contributing factor. And a single neuron represented by this line activates everywhere, right? So it activates towards infinity. So activation grow if you increase the distance from the ReLU neuron. So that was causing problem to me when I was trying to remove neurons or to add neurons, because if you remove something, it highly degraded the function. So I was looking into radial basis functions and metrics, because they seem to produce locally activating neuron. So I'll show you a visualization. So this is what I showed you right now. And I was also trying to approximate using radial function, and radial function gives a notion of distance or similarity. And if a data point is close to a center or a neuron, then it activates highest. And if it is far away, then it activates lowest. So that is how I was tackling it. But there were other problems, like if two neurons are together, then also it is difficult to disentangle which one is causing the output. So there was also need to disentangle the neurons. So I moved towards softmax that can create separation between regions. So I like to show this diagram. And what I also found is that distance is very easy to understand in terms of decision boundary between multiple classes. So top one is linear from the linear function, and bottom one is from a distance. So linear neuron is difficult to interpret because we don't have exact notion of which points that should maximize, because it maximizes at infinity. So what for distance it maximizes at the exact point that we are calculating distance from. So this is called Voronoi diagram. And this is the maximum softmax. Sorry, this is the like closest point around closest region around a point, and it is represented with softmax as well. Softmax gives some sort of smooth maximum, and that is what I am using next. So that is useful when you are creating dictionary learning. So is like you measure similarity between key and query, and if key matches with some query, then we outputs its value. It might seem a little bit difficult to understand, but we know we have studied attention. If you have studied attention, then it is more easier to understand. And softmax is useful to create something that sums to 1, and it has used in classifier layer as well to get probabilities of output classes. And yeah, what I got is I got something like this, the top one, right? So each is a center or a neuron, like here. This can be represented as a cross, and it activates in a local region, so that it doesn't activate where there is another neuron. The first problem was it activated in the region where far away from other points, like you can see here. It also activates well in some cases, but in some cases it activates in the faraway region as well. So that is a problem because we don't want to confidently predict what is not in the training set. And I think it is very important to reject such examples saying that the doesn't know in that direction. So what I found is we can use epsilon to give a threshold beyond which the confidence decreases. And I was able to create, here you see that with epsilon neuron, I was able to create locally activating neuron.
Hey, Suman, can I ask?
Yeah,
ask a
sorry.
question?
Yeah.
So can you remind us again, this is a network with two-dimensional inputs? Two input
Yeah.
vectors?
Yeah.
And in this case, you're training on this sort of Swiss roll data set or this data set, right?
Yeah, but it is just initialization
Okay.
to see where our neuron activates, just to analyze.
And what are so we trained the network, what are the ground truth labels you used?
Yeah, so one of the spiral is 0 and another spiral is 1.
Okay. The network you're showing us, the network we're interpreting right now, is it trained well? Is the training and validation curve, it's not working well yet?
Yeah. So this is just to produce locally activating neuron. We haven't gone towards training.
It randomly initialized, no training?
Yeah.
Okay. Got it. Okay, thanks.
Yeah. So I'll skip this math. Okay, so this may take a little bit long time to explain. So I'll get into figures.
Those softmax, yeah.
Yeah. Distance, you calculate distance and you negative distance and you calculate softmax. So whichever is the closest activates the most. And if you add epsilon to the softmax, then you can get a region that says it doesn't match to anything else. Similar to attention is off by 1. Yeah. So after adding the epsilon neuron, the neuron represents everywhere that there is no data point. And you can change the temperature of softmax. So there is a thing called softmax with temperature, and you can change the temperature to change the property of the neuron that activates in wide region or short region. And also, I was able to change the value of epsilon in the softmax to create something that looks like this. And I think this is very useful because when you are moving away from the centers or the neurons, you produce very low confidence, or the epsilon neurons win out. So you are saying that you don't know about the output because epsilon neuron is winning. You say, okay, you are not confident about the output. So if the epsilon neuron does not fire and some of the neuron fire, then you have a high confidence. And similar to these, like, okay, so these are just the neurons, like these are just the first layer neurons, and you successfully able to separate these neuron fires here. And what we can do is we can move this region and move such that we can make this data set separable. So you move the region containing blue class towards one side. So you move this region here, this region here, everything blue you move upwards, and everything yellow you move downwards. So this creates a separable transformation which you can easily separate, and also it is represented using interpretable neurons. So you can also classify to this data set with local neuron, and the epsilon neuron, this one is epsilon neuron. So if you try to predict something far away from the known data points, then it doesn't represent any class. So not 0 class, not 1 class, but this class is uncertain class. So it says yeah, you can reject such classes to reject adversarial examples, like I show the experiment next. And on with my experiment, what I found is that I was able to successfully reject. Can I go a few more minutes? Yeah, sorry. Yeah, so you can also classify, manually create the neural. You select all the classes and all the blue classes and move them towards a certain point, and you pick all the red points and try to move it towards uh, another point. And so you can separate these two classes. Also, sorry, you can also use the
Just for timing, we should wrap up fairly soon if we can.
yeah,
Thanks, Lim.
okay, so just 1 minute, okay? Just like what we did in 2 dimension, we can do in higher dimension as well. And instead of the crosses, we can choose data points itself as weights, and you can measure the distance between new sample and the weight, right? And you can, similar to this uncertainty diagram, you also get something uncertainty in higher dimensions, which you can use to reject adversarial examples. So these are the words, some of the sample of the words that we chose. And when we input some random input, the epsilon neurons fire the most, and you are able to easily reject it. And I also tried with adversarial examples with epsilon 0.5, which is we were able to reject the examples using the uncertainty measure of epsilon neuron. So 50% adversarial rejection. Also, it rejected some of the inputs. So some input were also rejected, but adversarial examples were also rejected. And when we allowed the weights to change very much from the data point, like that words doesn't look like one of the characters, then the property was gone. So you could not detect the adversarial examples. And it was crucial that the words looked like the actual data set to reject adversarial examples. So yeah, thank you.
Simon, thanks for presenting. This is really interesting. I think we should move on to the next speaker just for the sake
Yeah.
of time. But this is really cool work so far. Hope to see
Thank you.
maybe an update next time.
Yeah.
Thanks for presenting.
Thank you, Simon. Hassan, it's your turn.
Hello. So this is the previous presentation from last time. So it's just a quick replay. So can we generate our distribution DNA? What is DNA? Quickly, it is the genetic information which encodes all life, right? So it's encoded in A, T, C, and G. Obviously, there's other. It's not necessarily just the base pairs on their own. It's also the interactions between the base pairs, as well as interactions with the proteins that structure the base pairs themselves, etc. So sequences encode information, but it also forms structural support. Topology matters. A single base in the wrong location can have major effect on an organism's viability. Models must take into account both local and global information. Average sequence length for a species ranges from 5,000 base pairs to 150 billion base pairs, and the Holder exponent is 0.63. That is for a single species of virus. However, on average for viruses, the Holder exponent is around 0.58. And the Hertz parameter, this is the predictability, is 0.8. I've only done for a group of viruses. I've not extended for every type of sequence. So those numbers are just preliminary. So what's the current state of the art? You've got Hyena, you've got DNA BERT, you've got nucleotide transformer genomic pre-trained networks, so the GPN, you've got GeneLM, you've got Grover, and I think there's been a few since, so I've not updated the slide, but there's been 2. There's Evo, and there was another one that came out, I forgot the name. Most of them are transformer models, except Evo, which is a SSM transformer hybrid. So what's our available data? We've got the NCBI plus the nucleotide databases. So there are a bunch. There's the European, the Japanese, and I think the Chinese have another database. All of these are publicly accessible. They all have the sequences as well as the family that they come from, species name, etc. For plants, the average base length is 1.2 billion. For viruses, it's significantly less. It's 10,000 base pairs on average. Most viral families are covered. This is great for us for validating, so we can look at a specific family and then see how well can we predict that family if we train on all the other families in the viruses. So we have roughly around 3 billion tokens, and for eukaryotes, we have plus 10 trillion. So we've managed to scrape all the data now, and we have that saved online. How do we validate? So we've got spiders representation, H curves, C curves, color square, 2D line, and FCGR. So we've so that's the original plan. We've just stuck with color square, 2D line, and FCGR. So that's just a chaos game representation. So that's FCCGR, and the 2D line I'll show in a second. And the color square is square. And numerically, you can look at the chaos divergence of two sequences. Persistent homology, that is the intrinsic topology of a sequence, and then Wen's method is an amalgamation of the spider representations of a sequence. So this was the plan for the initial set of experiments: transformer, so train a transformer model, SSM model, and a wavelet across different parameter size, across different context windows, across for the viruses that we have, and then leave a family out, and then test how well we're able to predict the family that we left out. Closeness is compared to a simple model, that is the polynomial or the harmonic mean of a sequence. The model should be able to tell if a specific base is of lower quality. This is important for safety. That was again in the plan, but we've not managed to do that. This was the persistent homology experiment, similar thing. Again, we've not managed to do this, but I'll show you in a second. And scale. So this is for future. So dependent on the results from the initial scale, the best performing with the best loss and the best training regimen. Possible issues: safety, compute data, legal, ethical. Every single one of these can be their own set of slides. So if you have a model that's able to generate DNA sequences, there's a lot of harm that it can cause. Hence, we're not sharing the models or a lot of the more dangerous aspect. But the results will be shared. The models themselves will be shared amongst the scientific community, and those that we can verify that they actually are do need it for academic purposes. So that was the slides from the previous set of experiments. Now, quickly on to the results. So any questions, please feel free to ask, or if I'm speaking too fast, let me know. This is a Hugging-Face space that contains the results from our experiments. The space is still in progress, and we will be adding more models and more results over time, along with more representations. And this is for viruses, but once we've managed to finish our experiments for viruses, we'll extend to other type of organisms, so eukaryotes, bacteria, etc. Right? So let's quickly see a representation of a virus, right? So this is a random virus we just picked. This is the chaos game representation. Each one of these represents a particular KMA. So that is, what's the best way of describing a KMA? A KMA is a subsection of a sequence. So a viral sequence is just A, T, C, and G up to 10,000 base pairs. The KMA is in this looking at a 6. So we're looking at all combinations of a sixth that sequence. And if there is a particular KMA that's overrepresented, it's brighter, and it's darker in the sections that are not well represented. What you'll notice, on its own, it looks a bit chaotic, but if you were to compare it with a different virus, right, you'll see that the other virus has a completely different shape. So the astrovirus has a different set of KMAs to the Acta domestica denovirus, densovirus.
Can I ask, what are these two axes here, and what value are you plotting? I don't understand.
So each of those is representing a KMA, right? So a KMA is a subsection of the sequence. That is, and then, hold on, let me actually just show here. It'll make sense if I just show it this way. So we're looking at KMAs of length of 6. So that there is a chart of 36 by 36. So let me go to It's really finicky. Okay, I've got 6. Okay, so 6, count. Actually, I should be looking at the KMO. I was doing the wrong one. Yeah, 36. Okay, perfect. Okay, so this is all the possible base pairs, right? So it is a bunch of base pairs. This is the k-mers of length of 6. So these are subsections of 6 of the sequence. Does that make sense? And it's going to put that you'd end up. So if say if you're looking at a sequence of 4, and the alphabet for that sequence is 6, that means that set 4 can have 4 times 6. What do you call it? K-mers.
Wouldn't it be 6 to the 4th possible k-mers, but in reality, not all of those are represented.
Exactly.
And so that's what's shown on the x and y axis of your.
So in this, each one of them is a k-mer. So and each is a k-mers. The k-mer that's on the other side is just a reflection of itself. So that you end up with each is a k-mer. That's it. So it's the first k-mer, and then the second k-mer, and then the third k-mer, and the third and the fourth k-mer. And this is just these, and the density, the brightness of the pixels is just what do you call it? How many you have. So the more that you have, the brighter it is.
So it's sort of like co-occurrence. Is that correct?
No, it's not called. It's just does it occur? So how many k-mer do you have? The more of that specific k-mer, let's say AAA of A's, the more you have, the more than you have, the brighter that it'll be at this juncture, right? So let's say this, and same like with AAAT, that's going to be this. Let's say it's that one there. The more of that you have, the brighter it is, and each one is representing one of these.
Okay, cool. So it's kind of like a fingerprint for how much of each of these combinations is present in a given sample.
Exactly.
Okay,
Exactly.
thanks.
Yeah. Yes. That is the chaos game representation. The 2D line sequence is just so each. Let's say A is you give A a vector, you give T a vector, you give C a vector, and you give G a vector. And all of these vectors, you kind of just cumulatively summate them. You end up with a 2D line representation of the sequence. And in fact, I've got easy examples so you can kind of see. So A is that way, G is that way, C is that way, and T is that way. So every time, if there's an A, if that follows this A, it's going to go here. If there's a T that follows this A, it's going to go there. So and then carry every sequence that you have, right? So you can end up with a 2D representation. The color square in this instance is just each pixel point represents a particular. So you end up with 4 colors, each one representing a base pair, and you start from the top corner and you just keep looping around until you get to the central section. And if it's not a square, it's not going to fill the entire square. And you can kind of have a color square representation of the sequence. Different species have different representations. It might be difficult for those who are colorblind to kind of see because it's some of my colleagues are colorblind, so it was extremely difficult for them to see the patterns. But yeah, there is a bit. Persistent homology, again, it's just looking at the sequence and then seeing what's the different topologies. Wen's method is it's just looking at the spiral. It's a bit long to get into, but it's essentially you look at the spiral created by the base pairs. So each base pair can be a point in a spiral, and then you keep adding them up, and then you look at the distances between the two spirals, and you use that as a way to work out the similarity. So it's a neat way to data. So we did analyze the viruses, we looked at the underlying structures, and then we started the training of the models. So the first question we asked is, does context size matter for a nucleotide model? Does increasing the amount of some model sees if we control for the model size and the rest of the hyperparameters, does it actually result in a better model? Hopefully we answered that definitive yes, I think. But yeah, essentially, the more the model can see, the better the one. The second question is how do model types compare? So how does an SSM compare with a transformer? And for transformers themselves, how does a traditional transformer compare with a dense former? The dense former is just a transformer with the attention section swapped out with a dense convolution, right? So again, we compared so let's pick a particular model. In this instance, we can pick the Pythia, and we can parameter of 14 million. So we looked at the cross entropy, and then we looked at loss, that it's headless loss. That is, what if the model cannot know the sequence that comes before it? You just feed it 0, does it actually end up learning? It doesn't. Cross entropy performs best here. However, when we fine tune the model, the complement performs significantly better than the cross entropy, which makes sense considering it's just taking, you're looking at the base pair, and then you also include the complement when you're calculating the loss. Let's see, it's, yeah, you can, do we have, no, we didn't. Let's look at the complement.
Just for timing, we should probably wrap up in a minute or two. Yeah.
Okay. So again, we looked at scaling, which models perform better when it comes to scaling. Dense former performs significantly better than Pythia when it comes to scaling, even though they both learn. We found on downstream tasks that dense former is able to pick up better. And then we looked at the particular logics. Again, looking at particular logics, does a model learn better than if we were to look at the 2D representation, or cross entropy, or the complement. Yeah. So the space will be live, so have a look. We'll be adding more descriptions of what do each of these tabs mean, and the paper will hopefully be going on archives soon. And once it does, I'll link it back to the Discord. So yeah, with that, I yield my time.
Cool. Hassan, thanks for presenting. And thanks to all the presenters. Maybe join me in thanking everyone. The Fun Research Jam number 21. If you have questions for the speakers, please go to the Research Jam channel on Discord. And any of the speakers that have extra time, I'd encourage you to go there. Just say a quick hi so people know who to reply to or who to talk to if they want to ask questions of the speakers. Roseanne asked me to wrap up, so I think she's not here to say goodbye. But yeah, thanks to all the speakers, and see you guys next time.
Thank you, guys.